Hello everyone. Today we're going to talk about the cytochrome C comparison lab in biology, and I want to give you a brief discussion about the background and just an explanation of the lab so you can go ahead and get started today. First of all, we are investigating cytochrome C. Keep in mind that this lab is just another investigation into evidence for evolution. That's been our focus all week. And we're also going to continue our talk about how to create cladograms and phylogenetic trees for the last time. So this is kind of a culminating activity for everything we've been discussing all week. So cytochrome C is actually not a protein you've learned about before, but actually where it falls, which is part of cellular respiration, and inside the electron transport chain, you have learned about. In other words, where would we find this protein? Hopefully you're shouting in the mitochondria. And so think about the inner membrane of the mitochondria. That's what we're looking at today with this protein. And any organism that has a mitochondria and therefore needs to do cellular respiration would have this type of protein, cytochrome C, within the chain. So keep that in mind. Some DNA sequences code for mRNA, which, as we know, codes for the amino acid sequence of proteins. And so cytochrome C is just this protein involved within the mitochondria for energy use by cells. And it is found in most, if not all, known eukaryotes for that same reason. So you also should know by now, okay, if you've read the book, also you've seen that in the book as well. Over time, mutations in DNA sequences occur, and as a result of these mutations, organisms change over time. So this is only one protein we're looking at in the big scheme of things, but it has certainly changed over time. And so we're going to see that these mutations accumulate almost like a molecular clock as organisms change. So let's take a look at what you're going to do today. The method is explained below. I will let you know to save some time with this lab. You are not going to actually be doing what the lab will explain in methods, which is counting the amino acids that are different. Note in green right here, they are explaining how to do that. I would like you guys to read about that and just take a look at that situation, but there is no need to actually do so in terms of the counts because you will have the ability to just get these numbers off the data sheet that I show in a minute. So please make sure that you're looking at how to do this, but knowing that you're just going to be getting the data from a separate sheet to save some time because it is a very tedious process to count all these amino acids. So if we scroll down, they do explain for part B in the branching tree or cladogram how to look at the relatedness among species. And notice for species 1 and 2, they share a more recent common ancestor on the tree. And then it does tell you after that, if you have more species to place later, you would place the next 2 closest species on the next shortest branches. So as you finalize everything today by creating your tree, please don't forget about those rules you've learned. If we scroll down, you are not worrying about any of these data sheets or points with the amino acid differences. Again, those are highlighted. In the past, we used to actually count up those differences among organisms. And if you're looking at this like, holy cow, this is a gigantic protein. And so all of these letters actually represent different amino acids. The dashes represent places where we've had deletions. Remember, in evolution, it's just as common to see something change as it is to see something, be deleted. So a deletion is just another type of a change. If we scroll down, we can then see in table 1, this is the first place we need to begin, and that is going to give us the need to put in the differences between amino acids for 9 different species. So we are going to get that information over here, and that can be found in the comparisons table. I just pulled this up from the Google spreadsheet that I posted earlier today for you. So notice in this spreadsheet, there is a short form chart, there is a long form chart, and there's also the structure of cytochrome C. So everything is in one place for you. I would suggest that until you get to some of the analysis questions, if you would like to see more information and look at the long form chart, that's fine. But the short form chart is going to give you all of the information you need. The way this chart is set up is that actually everything above the darkened spaces is what you want to copy and paste right over to the chart that I gave you, because everything below is correct, but it's repetitive. So you do not need all of that information. So if we go back and take a look at this in Notability, all of these numbers then, right, you will need to go ahead and fill in from the information I gave you in the chart. Notice for everything underneath that is darkened anyway. You will not need to write those numbers in because although they're correct, they are repetitive. So please feel free to do that first and foremost today. You're just copying and pasting those numbers over. Once you have a chance to actually examine and analyze the numbers, please keep in mind, as it explains in the procedure you should read, these are not similarities in amino acids, these are differences in those amino acids. So two organisms, for example, like the horse and the donkey, that are extremely closely related may only have something like one difference, because that means all of the other hundreds of amino acids are the exact same. Therefore, that shows there is very much a relationship between those two animals. They are very similar. How do we know they're similar? Think about what you know about a horse and a donkey. Their phenotypes are similar. Their morphology is different, or is similar, and morphology is just the study of body forms and traits. So because all of the morphology and the phenotypes are similar, we say we have some background information on those animals and know that they must be similar in the proteins they hold as well. Once you have all your numbers filled in, please take a look at the chart below. Again, this is a branching tree, a cladogram, all right? Although we're expecting that if they wrote time in, it would be on the bottom and then progressing towards the top as we've seen before. So as you fill everything in, please take a look at the left side versus the right side. The left side is where organisms are very distantly related, okay, compared to those towards the top right, where we have relationships, if we go ahead and highlight this, that have just recently diverged as they share very recent common ancestors. I would suggest that you take a look at where you've had very few differences, and organisms that have had only few differences between them be placed then on the top right. As you move along to the left side, those are organisms that are more distantly related. In other words, more mutations have accumulated between them over time compared to those on the top right. And the other thing to mention is when you write these in, like in the Beasties lab, you will not actually be putting traits or tick marks or anything like that this time, because we're using biochemical evidence today for comparisons. In other words, DNA. DNA being universal is something that we understand. It's a great thing and probably the best piece of information for evolution because there are no gaps in that evidence like there are in fossil information due to the fact that not all organisms, as we discussed, fossilize. So like I said with the Beasties, you did something similar, but you didn't have to put the traits or tick marks in that lab. In this lab, because we're investigating DNA similarities and differences, you do not. All you would do is place the organisms towards the top of the tree right on the line as you believe where they belong. The bottom portion here, I'm going to circle this part. Okay, these are the only two organisms where their relationships are somewhat unknown because they're both so distantly related to everything else on the top right. And again, therefore, I really don't mind where you place these two. All right, you could revert them back and forth because we don't have enough information from just looking at one protein in the entire genome to figure this out. So I hope that makes sense. And when you are done with your tree today, the only thing I'm asking you to do is examine that and go to the Schoology discussion board that is posted in this day's e-learning folder and go ahead and make one comment. I'm only asking for one about two organisms, for example, in this tree and their relatedness based on this lab. So if you have any questions, feel free to email. You are not today responsible for the analysis questions yet. You will be making many predictions about this later, but today I'm not asking you to do that. This is going to be something that's likely going to be assigned on Monday. So I suppose if you want to work ahead a little bit, you could begin a few of those questions, but it's not recommended to do that if you haven't even read the book yet. I would recommend you guys go back to section 2 in chapter 18 and do a little bit of reading to feel better about this information, all right? Or go listen to some of the other screencasts again if you need to from the other labs that I have put out there, all right? I hope you have a wonderful weekend. Again, guys, please make sure if you have any questions, you email me and I will try to respond right away. I'm available directly between 9 and noon today, but I'm sure I'll be answering emails this afternoon as well. All right, take care and thank you again.